So principles of clinical chemistry automation. Like I told you previously, the clinical chemistry lab pretty much is automated. At North Florida, it's one big machine where you stick the tube in, and I got a video of a similar lab on here where it scans the barcode, which tells it what tests it needs to be run, then it sends it to a centrifuge, and centrifuges it, then sends it to individual machines to take samples of, the supernatant, and then test it for whatever was required. So it's kind of interesting. So the modern clinical chemistry lab, like I was just describing, uses a high degree of automation. And that's, anybody know why we want to do things automatically that were previously done manually? Yeah, because there's a lot of pipetting error that can occur from individuals, and if you have a machine that does it all the time, it should be, reduce that, what's that called? What kind of error is that? Huh? No, you got your systematic error and your, huh? I'm deaf.
I think they said random.
Yeah, like your random error. Which is what? Huh? Is that your accuracy or your precision?
Accuracy.
No.
I said the same thing.
Random error is your precision. That's going to be the size of that scatter around the target. Accuracy is how far the average is away from the target, right? Remember? Don't worry, I used to get those confused, and I still do all the time, unless I think about it for a second. So yeah, that's one of the reasons why they're going towards automation. It reduces your error. And this allows the operator to focus on tasks that cannot be readily automated, and increasing both efficiency and capacity. So this is like code for we need fewer of you, and you guys will do the things the machines can't do, right? So it increases the efficiency, meaning it costs that facility less. So, is that something different? Oh, yeah. So the analytical process is divided into three major phases. Your pre-analytical phase, and we talked about variations that we can observe in that pre-analytical phase. Your analytic phase and your post-analytic. So which phase are we talking about automating right now, do you believe?
Analytic?
Yeah, the analytic phase for the most part. So that corresponds to sample processing, or post-analytic will correspond to processing, chemical analysis, and data management, respectively. This is pre-analytic, analytical, and then data management, respectively, would be post-analytical. Substantial improvements have occurred in all three areas in the past decade, and the analytic phase is the most automated, which this was the answer to the question I just asked before, right? And more research and development efforts are increasing on automation of the pre-analytical and post-analytical processes also. So why automation? Basically, you want to increase the number of tests by one person in a given period of time. So you're increasing the productivity of each one of the personnel working in the lab, right? And it minimizes the variations in the results from one person to another because the machine's doing it all. Where are you going to get your errors in the maintenance of the machine, or sample labeling, things like that, or, the reagents that you have to put into the machine. Minimize errors found in manual analysis, equipment variations such as pipettes, and use less sample and reagent for each test. So automation really cuts down on the amount of reagents and samples that you're going to be using, so it can spread that sample out more, so you don't need as much for each test, and it allows for saving on reagent cost. And the goals of laboratory automation are basically reducing costs, right? Expansion of the laboratory testing to generate more revenue, so you want to reduce the cost per test, reduce turnaround time. What is turnaround time?
time it takes from when the test is ordered to when the results come out?
Yeah, so you want to reduce that, reduce the errors, and improve safety. So how's it going to improve safety if it's automated?
Less chance of cross-contamination, big blood on.
Yeah, exactly. If the machine's doing it all, there's less chance that you're going to poke yourself with the needle or contaminate yourself and so forth. They'll work with the nasty chemicals that some of these chemistry tests use, right? They're all back there in the machine, although you'll probably be loading the reagents and whatnot. So there's different types of systems. You have total laboratory automation, there's modular integrated automation, and then modular or standalone systems for front-end sample processing and archiving. So there's the three different types of automation. Your total laboratory automation basically works with this integrated track system that links all the workstations. Includes samples, sorting, routing, centrifugation, aliquot preparation, analysis, and sometimes post-analytical storage and retrieval. And I'll show you a little movie that I found on YouTube that has one of these systems, and they take you through all the different processes. They're huge systems. They take up a big room, and it requires major architectural work, far-reaching organizational changes to be effective in planning, and they're really expensive to purchase. They're pretty much less than truly turnkey, and they require a laboratory to select and coordinate equipment from many vendors. So they look kind of like this. There's this, a place, where is it? So you got robotic analyzer interface. This is the loading station that goes in here. The samples will go to this automated centrifuge, through all the testing equipment, and then back to be stored in a refrigerator. Huh? Push your button. Exactly. This is the little movie. Let's see if this works. How does it work? I gotta move it. I picked it because of the groovy music. So there's your sample. It's being loaded, and if you notice, it's getting dark. The scanner right there reading the barcode.
Ladies and gentlemen, this is the future of your job. I'm sorry, the life of your job.
And then it's going to be sorted to go to whatever machine it needs to go, right? This is on the centrifuge rack, so these ones are going to be centrifuged. It goes to this automated centrifuge. So it spins the samples. This is from a hospital in New Orleans that wanted to advertise that they just automated their lab. You notice it gets red all the time. Did you see any of the samplers yet? So you see that? It's like dipping down into a sample, or it's washing the pipette tip, and then it goes into the sample. Draw some sample up into the machine, and then it's going to wash. I can't tell which one is which, but it's doing one and the other. So what do you think an issue is with this where you're not changing pipette tips? Huh? Yeah, do you know what that's from? Contamination? What do they call that? Huh? Well, biofilm, but that's not the kind of contamination we're talking about. We're talking about if it goes into one sample, let's say it washes for a little bit and then goes into another sample, what's it doing? Huh? Yeah, cross-contamination. That's called carryover. And you can adjust the level of carryover by adjusting that wash time, right? Or the number of washes that it does. Does that make sense?
I don't have one where it changes the step every time?
They do have ones, but then that's an increase in what? Yeah, reagent and hard supply costs. So a lot of times they don't. So they'll just wash in between samples, and then you can adjust that level of wash, and that is going to adjust the level of carryover, right? But the more time you spend washing leaves what?
doing the
which is going to reduce what?
productivity.
Productivity or throughput, right? Those are the terminologies, but you get it, right? And that's a fine line that you have to come up with, is how much turnover, or not turnover, carryover are you going to allow to get the maximum amount of throughput. So that goes into with how much error is allowed, right? The amount of carryover you can carry over from one sample to the next. So how do I get back to this one? So, and this is just a picture of another laboratory automated systems model 7600 series, and I think these are kind of old pictures. Takes up a lot of real estate in a lab. Then they have these modular integrated automation, and it's developed as an alternative to total laboratory automation. They recognize the need to provide cost-effective analysis through continued consolidation of testing, but it's a little more flexible than a total laboratory automation, because you buy that all at once and it has to be used with the particular equipment. This one, it's modular, so you can buy one piece and then you can mix and match what it does, basically. It can still, they're linked by tracks or conveyors, so they look a lot like total laboratory automation, but they're not, they're modular. And they provide either analytical capability or pre-analytical processing, can be selected independently to meet the user's individual needs, and that's the whole key, it's a little more flexible, right? Pre-analytical platforms can be added to existing laboratories without disrupting the analytical capabilities of that laboratory. And we're talking, so pre-analytical are basically where you take the tube, right, and you identify, you enter it into the information system, scan it, and enter what tests it needs, or if you scan it, it does that. So that's like the pre-analytical phase. And they have those at North Florida too, I believe. So types of analyzers. So basically you can have continuous flow, where you have tubing that contains the reagents and samples continuously, and they're separated by little air bubbles, so I'll include an air bubble in that tubing, and you mix the samples in between the air bubbles, and then you'll run it through like a spec, and it measures how much absorbance or whatever is passed through that tubing. And that was like one of the original ones. Centrifugal analyzers, I'm going to show you a video where they do that they have these point-of-care chemistry machines now, where it's a whole chemistry lab basically on this little disc, and that uses a type of centrifugal analysis where you separate, centrifugal forces used to mix the samples and reagents. Then you have discrete analyzers, where separate testing cuvettes for each test and sample, and then random or irregular access type samples. So the continuous flow, it's the first auto analyzer, was a continuous flow, single channel sequential batch analyzer capable of providing a single test result on approximately 40 samples per hour. And analyzers with multiple channels can work, at the same time, produce 6 or 12 results, and produce 360 to 720 tests per hour. So it's a lot of throughput there, right? And in this continuous flow analyzer, samples are aspirated into tubing, introduced samples into a sample holder, bring in the reagent, create a chemical reaction, and then pump the chromogen solution into the flow through cuvette for spec analysis. And they have a little picture here where you have the bubbles that are going to separate things. Samples are separated by the air bubbles, and then you get the sample and the reagents meet here, and then it goes into the incubation and then the detector. But these aren't really favored because they have significant carryover problems, right? You can't do those wash steps like you can with the individual pipettes that they use in the discrete type analyzers. Does that make sense? So it's typically, and it's wasteful because they have a continuous flow of reagents too, so not so efficient. So it's used on some spectrophotometric instruments in which the chemical reaction occurs in one reaction channel and then is rinsed out and reused for the next sample, which may be entirely different chemical reactions. Centrifugal analyzers, you have discrete aliquots of specimens and reagents. They're pipetted into discrete chambers in a rotor. And then that's going to spin. You know, the specimens are subsequently analyzed in parallel by spinning the rotor and using the resultant centrifugal force to simultaneously transfer and mix the aliquots of specimens and reagents into a radially located cuvette. So the samples are in there, they spin, and the reagents and everything are added, and it all goes into the cuvette. And then the cuvettes then move through the optical path of an optical system, and you measure the absorbance that way. So a little bit better. And then discrete analysis is the separation of each sample and accompanying reagents into separate containers. They're capable of running multiple tests on one sample at a time, or multiple samples, one test at a time. They are the most popular and versatile analyzers and have almost completely replaced both the continuous flow and centrifugal analyzers. So in these, sample reactions are kept discrete or, by themselves, right? And they use separate cuvette cells, slides, or wells that are disposed of following the chemical analysis, which is going to increase the disposable cost, right? And this keeps sample and reaction carryover to a minimum, but increases the cost per test due to the disposable products. So this is basically a summary of the operations, right? First, you're going to have sample identification. That's done with a barcode, typically, right? And then determine of the test performed, and that communicates with the LIS. What's the LIS?
Laboratory information
system. Yeah, so that's the whatever facility you work at, it's probably going to have some kind of laboratory information system, which is software basically that communicates from when the order is taken or ordered by the doctor. It enters what test you want. Then the sample's drawn, that's on a barcode, so that code is matched to what tests are required of it. Then you take that and process the sample through the lab. And the LIS is going to know what test is going to be needed on that sample. And then chemical reaction is going to occur. The reagent systems and delivery, one or more reagents can be dispensed into the reaction cuvette. Specimen measurement and delivery, a small aliquot of the sample is introduced to the reaction cuvette, and the chemical reaction occurs when it's mixed and incubated. And then the measurement is done with optical readings typically. And the signal process gets from that optical reading will correlate to that calibration curve that was set up on the machine, right? And that's where we get that, what's the 10x rule violation, right? If the calibration was bad or something, you could have it shifted. And it sends the results back to the LIS, which will then, you guys would have to look up, and if it's outside of that normal or the reference range, you got to highlight it, right? And include all those reference ranges. And these are some pictures of different analyzers. Hitachi 902 analyzer. So with some automation, there's still some very basic steps, which is specimen preparation and identification. Again, labeling is critical, and programming of the instrument. Laboratory personnel are going to perform and observe the quality assurance and quality control, of course, right? So you're going to introduce the quality control samples the same way on every run. So how are you going to pick what kind of system you need? What is your workload like? Discrete, you do individual samples at a time, or you're going to do a large batch at one time. Single instrument or multiple instruments, you need to worry about storage of the reagents, right? You need refrigerator or freezing space because that costs more. Kept at room temperature until reconstituted. And then some concepts in laboratory automation is the test repertoire. So that's basically the number of tests that can be performed by an instrument at any one time. Random access is the ability to perform multiple tests on one sample of different combinations. So you could do multiple different combinations on one test with this random access type. Discrete is compartmentalized each test, so they're separated from all the other samples basically, or tests. Batch analyzer performs the same test simultaneously on all samples, so it's like a batch of samples, batch analyzer, right? Dwell time is the minimum time you need to obtain a result. Throughput, we talked about that. That's the number of samples you can get done in a certain amount of time. And then stat testing, what is that? No, it wasn't that. No, it's you know what that means. They need to test stat, which is like right away. So, and a lot of stat testing can now be done with point-of-care type devices. And I'll show you a little video of a new point-of-care type device too. So, and I was talking about point-of-care testing. What is point-of-care testing? So if we go through the whole process of patient, sample testing, you start off, patient and the doctor are going to communicate, then the doctor is going to send a request for some kind of test, which then goes to the phlebotomy department, and they have to go get a sample. They get the sample, they transport that sample, then they got to label it. You guys are supposed to label it before you leave the patient, right? Transport the sample, register that sample under the LIS. Then they take it to the lab where it gets prepared, do analysis on the sample, got to get the quality control done on the machine to make sure the analysis is okay, if the machine's okay. And then that validates that result. They come up with a report, transmit the report, which then gets interpreted by the patient or doctor again back at the communication, So what point-of-care testing does is it basically gets rid of all of this, right? Everything down below. So basically, point-of-care testing, you could do it right there in front of the doctor. They get the results right away, and then they can communicate, the doctor can communicate what that means to the patient right there. So it saves a lot of time, basically, right? You think it's going to save on reagent costs? No, yeah. So,
point-of-care testing.
yeah. So the places you can do point-of-care testing is basically anywhere, right? Physicians' offices, they do them often. These chemistry machines that I'm talking about that are point-of-care typically done in not operating rooms, emergency rooms. But you can do stuff in operating rooms too, so you don't have to, transport the patient out of, or not the patient, the sample out of the OR. You can get the results right there in the OR. Intensive care units, home health care, and patient performed. We know diabetics are constantly monitoring their blood glucose levels, and that's point of care also. So, and it's defined by College of American Pathologists, analytical patient testing activities provided within institution but performed outside the physical facilities of the clinical laboratories. Okay, and alternative names could include bedside testing, ancillary testing, or off-site testing, and waived testing. Although you can have point-of-care testing that's not waived. And do you guys know what that means, waived or anything? Probably not, because I haven't talked about it yet, right? Only if you pre-read the book, maybe. So point-of-care testing, CLIA 88, we've talked about that before, and it's basically Congress passed, it's the Clinical Laboratory Improvement Act. They passed this law in 88, or this act anyways. But the regulations weren't written until 1990, and it wasn't implemented until 92. And it's overseen by the FDA and the CMS. What's the CMS? Central Mechanics Services? I don't know. I don't know what the CMS is. That's something I didn't look up. Nobody knows? And you always look at everything up. Oh. Sorry for not knowing things. So point-of-care testing, typically laboratories are going to be, they're classified as to what kind of tests they can perform. You have those that perform waived tests only, and we'll go on to what waived tests are. Those that are performing moderate complex testing, which has includes allowances for physician-performed microscopy. So you guys taking the hematology? Some of those would be more moderate or complex tests, because you got to sit there and identify what cells are what, right? So that takes thought on your part and an education on your part to be able to identify different cell types. So that's not something that's going to be waived. That's kind of highly complex test, right? Classification is based on the difficulty or complexity of the test procedure and the level of training required to accurately perform the test. So it's to be able to identify the cells and count them. It's kind of a complex test. Laboratory standards differ from each of these categories. So for the waived test, you don't need you don't need to be in an accredited lab, basically. No certification requirements for those performing the test, right? The lab must be CLIA, they must have a CLIA waiver certificate. No proficiency testing is done on these labs, no minimum training for the test personnel. Criteria for a waived test is if the results of the test are in error, there's not going to be any immediate danger for the patient, basically, right? So that's why, you can take them home and do it, they're approved for that, so there's no danger if you get a bad result. Like it says my blood glucose is 800, but yet I'm not in a coma. So if that was an error in the result, right? Examples of some waived tests, there's several methods of hemoglobin determination. Blood glucose, we've all seen those, right? What's HbA1c?
Your A1C is your residual glucose in your blood.
Yeah, it's glycosylated glucose, or glycosylated glucose, that makes no sense. Glycosylated hemoglobin, so that's where glucose is chemically reacted with the hemoglobin. You have glucose physically, covalently attached, basically, to the hemoglobin. Fetal occult blood, spun hematocrit, have you guys done that in hematology? So they're moving away from that because there's a lot of error in doing that, right? Ovulation tests, urine pregnancy tests, urine dipsticks, ESR. Oh. What is ESR? I looked it up today.
Well, looking that up, CMS is Centers for Medicare and Medicaid Services.
Is what?
Center for Medicare
Yes.
and Medicaid.
I should have known that, because if you violate these proficiency tests, the center, you won't be able to be reimbursed for Medicare and Medicaid.
ESR is erythrocyte sedimentation.
Yes, I knew that too, because I looked it up today. But I forgot because I'm here now. And what do you think PT is? No, physical therapy. No, you're right, goes on. Cholesterol, aminoacids for mononucleosis, and then they have these are pretty cool. These Pico Color Press, where is mine?
Test for mono?
Yeah. Or it's a waived test. I don't know. You might be able to buy it at your pharmacy. But they have these. They're like these little discs. Look at all the tests you can do. I think if I go, where am I? Here. Here's the detailed ad.
Very detailed.
So it's this machine, it's considered point of care, and they have all these panels you can do. They have this comprehensive metabolic panel where they test for ALB. What do you think that is? Huh? Albumin, ALP? I think we're going to do an ALP test this semester. ALT? ALP is alkaline phosphatase, right? ALT is... I think this is aspartate transferase, or aminoaspartate transferase, alanine transferase, BUN, blood urea, nitrogen, calcium chloride, CRE, creatine, glucose, potassium, sodium, total bilirubin, carbon dioxide, and TP. We just talked about TP, right? No, we talked about PT. What's TP? I don't know. I should know. But you can do basic metabolic panel, lipid panels, lipid panel plus, liver panels, general chemistry 6, general chemistry 13, all these different things, all on one little disc, which is pretty cool. This movie here, I think, will it click to that? Huh? Yeah, well, they're expensive to do, but then you could do it right there and you get your results. So this is an example of doing it. Here's one of the little discs, and all you need is 100 microliters of blood, whole blood. And this is what they're going to do. They're taking capillary blood for this test, which is going to resemble what, arterial or venous more as far as, well, for maybe oxygen, but I think for metabolites, it resembles venous. Huh?
Bad spot.
You're not supposed to milk it?
Yep. But it's hard to fill up one of those capillary tubes, isn't it?
Yeah, that's why you just do a tube.
Yeah.
I have a question.
Yeah.
If you, in a job, so who draw, do we draw the blood or the doctor draw it? That's kind of an question.
You'll have to draw blood sometime.
Phlebotomist does in the hospital.
Okay, so I'm not drawing the blood.
No, you guys got to learn how to draw blood.
Yeah, you do have to be able to do it. You have to be proficient. Matter of fact, when you do blood banking, you have a week of phlebotomy you have to do in the hospital.
Yeah.
Which includes geriatrics, bariatrics, neonatal, I don't think you do that.
So look at this. They just put it's hard to see, isn't it?
So we have to.
You see that blood filling it up, the channel?
I see it. Little circle.
And now he's showing us a semicircle in there. So he puts it in the machine, and then it's going to spin it and separate the sample out to the different tests.
I don't like having that figured out.
I don't either. Every time I get blood, I always tell people, I'm like, this is my least favorite part when you poke my finger.
And then you get the results there in like 5 minutes, I think, of all of those. Huh? Capillary? I hate taking blood. I hate bleeding. It seems like I injure myself every weekend.
At first I wasn't, and then when I asked Dr. Smith if I had to, he was like, oh yeah.
At first you want what?
John and I have to do IV bleeding time for hematology. And I didn't really want to do it, and then Dr. Smith basically told me that I had to, and he sounded really excited about it. And then he told me that I had to do Rich.
Oh, yes.
Then I told Rich, and he was like, I guess I have to get the really deep lancelets, and I was like,
Yeah, we so the lancet we have didn't work on my hand. Like we're sitting there.
Too many calluses?
Me, Willie, and Rich were all in there, and I have to get a large bore, and I won't put the large bore.
I hate doing that too. But oh well.
I didn't tell you about watching somebody bleed for like 3 minutes and stuff.
Why do they got to bleed for 3 minutes?
IV bleeding time. You have to like put a blood pressure cuff on, and you do a deep, it has to be 1 millimeter deep. I know, but I have to.
Don't even say later.
So I have a question. So if we were working at the medical technology, will be the one drawing the blood and running the test on them?
Depends on what department you're in and where you're working. Like we had one of our graduates she's a tech now at the hospital in Williston. So they specifically picked, she was a good match for that job because she was really good at phlebotomy. So she does it a lot of herself because it's a really small hospital and they don't have a lot of phlebotomists.
They don't have a blood bank.
Yeah, so she's a good match for that. But if you go to like North Florida to the micro department, you're not going to be drawing anybody's blood. Or if you're in the chemistry department, you'll just be putting the tubes on the and doing the quality control and quality assurance. So, but if you're in blood banking, you might be doing some blood draws. So So moderately complex, the lab has to have a certificate of accreditation. And it includes many or manual procedures with limited steps in preparation or automated analysis that do not require operator intervention during the analytical process. To get the accreditation requires proficiency testing. We talked about that a lot, right? There's CAP, was one of the places you can get proficiency testing. There's another one on here that I list that I forget their name, and I always do. And I looked it up again today, and I forgot it already. CAP is the College of American Pathologists. But then the other one, you'll see it when it pops up. So personnel have specific training and orientation to perform the test. Continuing competency must be documented also. And they have to have at least a high school diploma to do it. And then lab must have a lab director and a technical consultant to be accredited to do moderately complex testing. Highly complex testing, again, you have to have a certificate of accreditation. Must meet all the criteria for the moderately complex testing lab. Personnel, however, have to have at least an associate's degree or equivalent. And unlike the moderate complex level, the lab has to have a director, must have a director/technical consultant.
So when it comes to associate degree, are you talking about a bachelor's degree or what kind of associate degree are you supposed to have?
So they have, well, if you have your bachelor's degree from this, you're beyond the associate's degree, so you can have a higher level position. You can be a...
Wouldn't it be like the MLPs, those people that go to
Yes.
technical schools?
Yeah, the technical school associate degree type deal. You're right. So.
They have the program in Ocala.
Yeah.
And it's like, but there's no jobs in Ocala.
Really?
Yeah, there's not, I think MLTs, like, there's 2 hospitals in Ocala and use them and they don't have a lot because all they have is NPs.
Yeah.
So it's kind of like, yeah, we'll certify you, but you won't have a job.
You have to move somewhere, basically. Highly complex testing can include procedures that require considerable decision-making, like, your counting, right? Specialized scientific and technical knowledge. Tests that require special preparation of reagents, extensive technical and quality control measures, like HPLC, MS, and complex judgment and analysis of the results to provide interpretation, which is looking at, like, cell samples from, fluid preparations from patients. And most hospitals perform moderate to highly complex testing and must have hiring guidelines, participate in proficiency testing, and maintain comprehensive record keeping. Have established quality assurance programs, so you ensure that you're going to get good results, and are subject to government inspections also. So proficiency testing is mandated by CLIA for all labs performing moderate or high complexity testing as a method for evaluation performance quality by an external agency. And we talked about this, right? Labs are provided with samples of an analyte. Values are known only by the testing program, and then the lab analyzes the specimens the same manner as patient samples. And then they get the results back. There's a group mean and group standard deviation. They get their Z-score. And you're only allowed so many Z-scores until you fail. If you do fail on a particular analyte, you'll get another chance to reanalyze it, hopefully to prove its quality performance. Because, right, statistically, you might have an outlier out there. If it fails that again, then there's a possibility it will not be approved for reimbursement from Medicare and Medicaid for that analyte, right? So that there's their Medicare and Medicaid. So program requirements, appropriate CLIA license, chosen inspected certification agency that can be CAP, right? Support staff, standardization, structure of authority, responsibility, and accountability, interdisciplinary communication and relationships, and for point-of-care testing applications, the highest volume is typically what? Glucose, right? Likely to be the first application for a point-of-care test, home testing. Instrument becomes very sophisticated and maintain a log of QC results. You want to do that, right, if you're in a facility. You want to have your QC results in a log of them so you can tell when the machine needs to be tossed. Chemistries and blood gases, coagulation testing, activated clotting time for monitoring heparin therapy, hematology, spun hematocrit can have several opportunities for error, and this is what I was saying, it's being replaced by just specific hemoglobin measurements instead of spun hematocrit. You're going to have point-of-care connectivity, so a lot of the places in the facilities will communicate wirelessly with your LIS, your laboratory information system. So you get the point-of-care results, you scan the patient's wrist or whatever, do the results, and it goes right into their laboratory information system, their file that gets their results and everything. And that eliminates error due to transcription results. Most instruments are now capable of electrically sending the data. And I don't know if that's the most recent development in point-of-care technology. 3 components to connectivity is the device itself, the instrument itself can store the data. Data management system, it uploads the data to a workstation or interface where the data manager transmits test results to the laboratory information system. So personnel issues with point-of-care devices is most often they're performed by people who aren't laboratory personnel, where it's still the laboratory that's responsible for all the point-of-care testing. Does that make sense? So what do you think you have to do? So there has to be somebody designated as being in charge of point-of-care testing from the lab, and then they have to be in charge of training everybody that does the point-of-care testing on the proper way to collect specimens and do the quality control and everything, right? Does that make sense? And then they have to do reoccurring training and keep the machines up to date. So it's most often performed by non-laboratorians, physicians, nurses, nurse aides, respiratory technicians, not specifically trained in the requirements of accurate testing and interpretation. But the laboratory is still responsible for results, so they're responsible for the training and management of the point-of-care testing programs. Must build a structure to support and facilitate the point-of-care testing. So you're going to have a director, and the director, a laboratory director has to have a PhD or MD, laboratory scientist or a pathologist, and a point-of-care coordinator, which can be the laboratory scientist with a high level of technical and interpersonal skills, right? You got to have those interpersonal skills because you got to interact with all the physicians and everybody that you have to train. And the point-of-care trainers can be any designated person, for problem solving, etc. Training should include basic principles of testing, demonstration of proper use of the system, consequences of improper use, so you can give examples of what happens when you do something wrong and how that's bad and how to identify when you've done something wrong. Instructions for sample collection, the importance of complete documentation of all results, appropriate calibration and QC testing, and practical experience to satisfy the instructor that the trainee is competent. So what can go wrong? Something wrong with the equipment. Daily checks and documentation should be able to tell you about that. Operator technique, and that goes to training. Proficiency testing will pick up on bad operator technique, right? Sample unsuitable for testing on the device, and that's from poor sampling or limitations of the device itself. And the point-of-care testing operator takes responsibility for all these. Make sure you follow the standard operating procedure. It's there to protect the patient and you guys. And this is the other group that gives proficiency testing, the JCAHO. Any ideas? I looked it up today, but I forgot it already. I forgot everything today. I got things I should be doing today.
Joint Commission Accreditation Healthcare Services.
Yeah, that's it. Joint Commission. The Joint Commission and Accreditation Healthcare something. What's O? Offices? What?
Operators.
Operators. So
I
And
do
they-
with organization.
And these are their like standards. I'm not going to test you on their standards, but just so you see what accreditation agencies are looking for. So.